Okay, now tell me, what do you understand about the lipids?
Lipids are fats and oils.
Fats and oils. Any more? Anything other than that?
To break them down, you need the enzyme lipase.
Hmm.
That's it. I
We
don't
need
know.
the enzyme lipase. Okay. Okay, good. So, when we talk about the lipids, always remember that we the lipid is another word for the materials which are greasy materials. The materials which are greasy, slippery. The important part is the lipid is a part of our body that is very essential. Always remember the what happens according to the previous discussion. I would like to tell you that I sometimes feel like uh, the whole world may have done some kind of wrong propaganda against the lipids. Like
Yeah, because
they
everyone
have
is
made,
told that you shouldn't eat fats.
yes, everybody is saying negative about the lipids or fats. Remember, always remember, if you ever happen to listen to this lady, uh, she's Australian, Barbara O'Neill. According to her, and it was a total coincidence that due to constant reading, I came to a conclusion that lipids are the dietary lipids. Dietary lipids are never dangerous. Never dangerous. The whole world is basically working. The whole world is basically following some false concepts about the people who are basically shredding their body. Shredding means they are trying to make cuts on their body, and they are trying to show off their body. When you try to show off, you need to cut down the fats. That is a reality. But always remember that dietary fats, the fats that you eat in diet, are healthy. The things which are extremely dangerous, if you want to listen to any particular scientist or a lecturer about the diet, follow her on YouTube. Anyways, I'm
Okay.
coming back to the point.
Yeah.
uh, Anything that is one of the most dangerous thing that we often use and we don't even try to avoid them is the refined cooking oil. We don't even try to avoid them. You know what do we avoid? We avoid the buffalo meat, we avoid the bull meat only because we falsely, we mistakenly think that they are stuffed with the cholesterol. No, it's not right. This is the type of oil, the cooking oil, that is most dangerous. And nobody even talks about it. Another thing that the people are getting so wrong, they often use the banaspati ghee or the ghee.
Just
The
desi ghee
ghee
or
in there. Desi ghee is excellent. Desi ghee basically is extracted from the milk, which is an animal fat. It would be categorized under the dietary fats. And dietary fats, as I have already told you, are almost always healthy. You know what is a banaspati ghee? Banaspati ghee means that there was an oil and you passed it through the process of hydrogenation and you made it a ghee. What is the difference between a ghee and oil? I'll tell you that more openly. But always remember, the oil is considered to be liquid at room temperature, and the ghee is you can say semi
Yeah.
solid at room temperature. When you make an oil artificially in the industry, you convert it into ghee. This is what the most dangerous thing is. So every time,
Sorry,
whenever there is a
sorry, 1 second.
Mhm.
That word hydro, how do you spell it after?
Hydrogenation, adding hydrogens into the oil,
Hydro.
hydrogenation. Always remember, if there is a word added to anything, everything would be dangerous, and that word is refined. Everything is dangerous in that case. Refined sugar. You know, I have totally boycotted the uh, the sugar. Totally. I eat nothing that may involve refined sugar. I'm not asking you to do it. I'm just telling you, I'm aware that what is the consequence of this. Always remember, you may eat fruit. You know, the type of sugar, the type of carbohydrate, or the type of sugar that the fruit contain, and the type of sugar that is present on your table is same. And what is the name of that sugar, if you remember? The table sugar.
Is it refined?
No. Sucrose. From the previous class, I told
Oh, I
you.
thought you were saying the yeah, sucrose.
Yeah, that is refined sugar, but the nature or the chemical name is sucrose. Now, imagine I am, I being a person who has done the research, I am totally avoiding or even boycotting the type of sugar that is present in on my table, but I eat the same sugar in the watermelon, in the mangoes, in the oranges. Why? Because the sugar that is present on the table is highly concentrated, extremely concentrated. You have made the crystals of it. And whenever you eat the mangoes or watermelon, there is water inside those fruits. There are vitamins inside those fruits. There are fibers inside those fruits that would help you a lot. And the amount of sugar is very less. That is why I don't even touch. Now it is my habit, and this habit is very, very difficult to make. I don't touch the biscuits because they contain the same sugar, refined sugar. I don't take the cakes.
Yeah.
Everything that contains this, either refined oil or refined sugar, is not recommended. The thing that should have been recommended are dietary lipids, but due to propaganda of the industrial ghee and oil makers, we are avoiding dietary lipids and taking more dangerous oils. That is the phenomena we should be trying to understand. That is total propaganda. Like, there is a particular terminology. One terminology is beef, and there is another terminology called as cara beef. What is cara beef?
And I don't know, I've never heard of Caribe.
The beef is basically the either the meat of cow or a bull. And the cara beef basically is the meat of a black bull. The black, or you can say male of buffalo. There's a difference between a buffalo and a cow. So, the cara beef is a type of meat that contains way less cholesterol than the beef. If a person wants to avoid more dietary fats, he should be eating cara beef. Cara beef is very helpful. And like over here in Pakistan, since the Eid is coming, over here in Pakistan, we are readily available with the black bulls. So we just we sacrifice them. So which is acceptable, obviously. That is why we do that. So, but they contain less cholesterol. So remember that the lipids used to be a very notorious group, but they are not. Why? Always remember the 50% weight of your brain is fat. Your brain is not merely fat, it's cholesterol.
So how much percentage is cholesterol in the brain?
50% of the weight. Remember,
Oh, of
I'm not
your
saying 50% cells, 50% of your brain's weight.
okay.
Like if your brain has some weight, basically that weight is because of the fats. Otherwise, there wouldn't have been a weight of the brain. So, but one of the most important part about the cholesterol or fats in our body is involving the cell membrane and maintaining their structure. Your cell membrane cannot maintain its structure other than without the cholesterol. So just imagine how much important these are. There is another important thing in our body that is called as adipose tissue. Adipose tissue is a type of tissue that stores the fats. And the storage of fats, basically, the storage of fats, it acts, the adipose tissue acts as the body's connection uh, connection fibers. What does it mean? If you want to, if you want to uh, keep your body tissues and body parts connected to each other, you need the fats. The fats basically, the fats basically are acting like a glue. The fats
What?
basically are
Like
glue,
glue. Okay.
glue. The fat is basically acting like a glue, sticking together, sticking different parts of your body together, sticking different tissues together. So everything is important. Sorry, everything is glued by some of the proteins and many types of many you can say layers of fat tissue. So that is another way of telling the importance of the fats in your body. Okay. Next, how the lipids or fats are made. That is important. Lipids chemistry. Uh, the main basic chemistry is same as carbohydrates. CHO. But remember, the fats or lipids have very less oxygen content. Very less oxygen content. So more, it means more carbon and hydrogen bonds, and they have almost double energy or even more than double energy compared to carbohydrates. But the problem is they are very difficult to break, as I may have mentioned this already, that they are very difficult to break. So that is why we don't often use them for energy. But if there is lessening, if we try to if we don't provide the body with the fats, with the glucose or with the sugars, our body would start breaking the stored fats. Or it would start breaking the stored fats. Suppose that a person is on a diet, and the person is, and that person is basically avoiding the sugars. What would happen? That once you avoid the sugar, your body would try to get the energy. Since there is no energy, since there is no particular energy in the food, now your body would take the energy from the stored fats. So that is basically what the meaning of the whole energy thing. The primary target of our body is to use the sugars as the energy. But since if you don't provide it the sugars, if you don't provide the body with the sugars, now you would have to get to the fats, and that is where you basically start losing the weight or start losing the fats.
Okay.
Okay. So next, the basic another type of when we talk about the chemistry, when we talk about the lipid chemistry, how the lipids are made. Always remember that every lipid compound has two features in common. Number one, it has a fatty acid in it. Number one. Number two, they are hydrophobic. Mostly they are hydrophobic. What is the meaning of hydrophobic?
Like, I'm assuming it would literally mean fear of water. So
Fear of water,
that
yes.
so it won't have water in it. It doesn't have
It
water.
would not be like, it would not go along with the water. It would not go
Okay.
along with the water. It does not mix with the water. It would keep its own identity. It would keep its own identity inside the water. Have you ever seen the drops of the oil inside water?
Yeah.
How are they? How are
They
they like?
float.
How
They
they look
f-
like? Sorry.
they float. So could you
Exactly. They
say
would not be mixing.
they don't like that when oil is in water, it doesn't osmosis doesn't take place because it's hydrophobic? Can you say that?
That's a very genuine point. Yes. When you would uh, use the fat somewhere or lipid somewhere, the water would not be able to cross it. That's a very genuine point. Now, try another thing. Try another concept. I have said a few moments ago that the fats and lipids are essential parts of our body, our cell membranes. So if they are essential parts of our cell membranes, then how the water cross the cell membrane? You got the question?
So how the lipids cross the cell membrane?
How the water would cross the cell membrane if cell membrane is made of lipids?
No, because the cell membrane's not mixing with the water. It's just letting it pass through.
Suppose that you have suppose like a drop of oil in the water. Uh, the inside of that drop of oil is the cell. Suppose inside that drop of oil is the cytoplasm. If you add, if you want to move the water into the cytoplasm, how would you move it?
I didn't get the question.
Remember that. Okay, okay. Just ignore it. Just for now, for the time being, remember remember that the lipids are hydrophobic and they are always made of fatty acids. And there's a rule. When you add fatty acids to any compound, it would become lipid. It would become lipid. Suppose there is a compound that looks like this. It has 3 branches. This compound is called as glycerol. When you add 3 fatty acids with it, suppose these 3 lines represent 3 fatty acids, this compound would now be called as a lipid. This part is glycerol, and the 3 fatty acids, these are fatty acids. When the 3 fatty acids would bond to it, it would now be called as a lipid. So that is how a lipid made. Remember, every lipid has to contain fatty acid. Without fatty acid, you cannot imagine a lipid forming. Now, the lipid can be of 2 types. If they are solid at room temperature, you would call them fats, and they are mostly from animal origin.
And if they're liquids, then oils.
Yes, animal origin, mostly from animal origin. Even desi ghee is a semi-solid. I can give you an example of it. There is after some time when the milk cools down, after some time there is a sticky or greasy layer above the or you can say at the top of the milk. Have you ever seen that? There's a
Yeah.
greasy layer appears above the milk. That is basically the fats that are like due to low temperature.
Is
They
it the
are,
malai?
yes, they are becoming semi-solid. They are basically the fats. But remember that if they are solid or semi-solid, it depends on the temperature. If at room temperature, they are solid or semi-solid, they are definitely fats. So whenever you see the thick greasy layer, which we call as malai, it appears above the milk. That malai is basically the fats trying to get semi-solid at room temperature. When that layer appears when the milk cools down, isn't it? When the milk cools down, it means that when the milk comes to the room temperature, its fat would try to become semi-solid. So that would be fats. And the Eid is coming. Like, in the country you live in, do you do the sacrifice?
I don't think it's allowed actually. Illegal.
Mhm. So, like, what happens basically that when you cook, especially when you cook the beef, uh, and you put it in the refrigerator, it becomes when you if you take it out after 4 to 5 hours, there is an orange layer that appears above the curry. That orange layer are the solidified fats. But I doubt you may have seen that. So then there is like liquid. If the lipids are liquid at room temperature, they would be oils, and they are mostly from plant origins. And you know very well that whenever we talk about the oils, they're like mustard oil or olive oil or
Yeah.
these kinds of oils, they are from plant origins. So that is what the lipids are. Okay, so done? Clear?
Yeah. Finished writing.
Hmm. I'm opening the new Jamboard because it was completed. Oops.
Can you send me the link, please, for the
Link of the previous one?
no of the new Jamboard?
New one, it's in the message box.
Oh, it hasn't loaded yet. Just 1 second. While I wait, you can just start. I'm anyway viewing your screen.
Sorry?
I'm viewing your screen anyway, so I'll just wait for it to load later.
Okay, yeah, sure. So now a very important chapter is coming, and this chapter is called as the food tests. This topic, not chapter, or tests for biological molecules. For biological molecules, and maybe yes, these are basically this is the topic. So what happens basically, we have whenever we try to test something, we have two kinds of tests. One are called as qualitative tests, in
And
which we
quantitative.
see the quality of a solution. Suppose change of color, and maybe the changing in the consistency, or the you can say viscosity, and maybe change in the um, yes, precipitation, maybe precipitate formation. So these are qualitative tests,
Then
and
there's quantitative.
yes, mostly food tests are qualitative. Why? Because we mostly rely on color change. We mostly rely on color change. The quantitative tests that we often um, do, these tests basically are, you can simply say that these tests basically give us proper readings. So we have more standardized results, reliable results, because we are actually we are actually getting the reading on which maybe the reaction is happening. So in the food tests, we have some general principles. One is the original solution. The original solution would be a solution containing solution that is containing the molecule, like carbohydrate, protein, or fat. A solution that is containing the molecule. Then we have another solution that is named as reagent. Reagent is a, a chemical that may react with the original molecule irreversibly, changing its properties, but not converting it into products. Converting it into products. Suppose I have a small example. You react the glucose with a reagent called as Benedict. I'll tell you the detail later. Now the
solution?
Benedict solution contains the copper ions. Basically, it contains the copper sulfate. That is why it is poisonous. So the glucose basically attaches the copper with it, and the reaction happens. The color changes, but now you cannot use the glucose again, and the glucose is of no more use to us. We cannot convert the glucose into any other thing. No useful products are formed. But it would show us a color change. That is why we call them reagents. See, reagents and the reactants both are different things. Reagents are those which do not convert them into products, into useful products. But the reactants and other chemicals, they are different. They convert them into useful products. The reagents are not enzyme. They would not continue the reaction. So that is what a reagent is. Got it?
Yes.
Okay. Next. Now, whenever we use the reagents, we rely on color change. That I have already told you. We rely on color change. The reagents that we often use, one is called as Benedict's reagent. Now, the Benedict's is because it was made by a person named Benedict. Then there is another reagent called as Biuret. Biuret's reagent. And then the third reagent is simply ethanol. The third reagent is simply ethanol. So, simply saying it, you can just imagine that these three different reagents are going to be giving us the identification. And the composition, currently, what you need to know, the composition of these reagents, the ethanol, you know very well, the ethanol is C2H5OH, but in the Benedict and Biuret, one thing is common, and that is copper. Copper. Okay. So, and Benedict is for, at your level, we would say the Benedict is for glucose test. But it is a truth that Benedict is actually for reducing sugars. But obviously, we don't need to explain what are reducing sugars because it's not required by our syllabus. Biuret is for proteins. Biuret is for proteins. And the ethanol test, the complete name of this test is ethanol emulsion test. Complete name of this test is ethanol emulsion test, and this ethanol emulsion test is for fats. Benedict, this test is Benedict test. This test is Biuret test, but the name of the last test is ethanol emulsion test. Emulsion test. so in which we would be using ethanol to identify the fats. Got it?
Yes.
Okay, so now one more thing. What is a positive test?
That I think it's like when you have certain hypothesis, and if the test and if your hypothesis comes true after your test, then it's a positive test.
Yes, you can say it follows the expectation or follows the hypothesis. That is a positive test. And then there is a negative test. Obviously, it does not follow your expectation. It does not follow your hypothesis. And
Wait, but
one last thing. Mhm.
I understand what this means, but like, isn't it then subjective? Like, for example, a COVID test. So, expectation is a bit like should be something else, right? Like hypothesis instead.
Yeah, yeah, exactly. Expectation should be replaced with hypothesis. I was going basically at a very small level, so I wrote the expectation. Obviously, it's hypothesis. So then there are some tests which are semi-quantitative. Semi-quantitative means that there are multiple color changes uh, representing the intensity of concentration. Suppose when we do the Benedict test, we get green, we get yellow, we get orange, we get red, and we get brick red. Now the intensity of the color would mean, this means negligible. Negligible, and we consider this test as negative test. If the Benedict ends up in green color. If it is yellow, there is sugar in the solution. Orange, red, brick red means there is a very high concentration. This is called as semi-quantitative. Semi-quantitative.
What
Like
makes it
you,
semi?
semi means that you are getting the color change, which means which is a qualitative property, right?
Yeah.
Color change is a qualitative property,
Okay.
but since you have the shades of color representing the intensity or representing the concentration, representing the amount, so that would make it semi. You are not getting any number, still you are getting the intensity. Getting the idea of intensity without quantity. Without quantity. That is semi-quantitative. Clear?
Yes.
Okay. So now I'm about to send you a particular table that would help you a lot in understanding different kinds of tests. Identification key. Just give me a moment. I would be. I'll check this particular document. Maybe there is a change I have to make. Red, orange, red, brick red. Okay. Pale violet, blue, purple.
Are we gonna do it till 9 o'clock or 9:15?
9:15.
Didn't we agree that when we're doing 3 classes a week, we'll do it 1 hour? Just
No, Sir Asan said that we should be going on with the
okay.
uh, 1.25 hours. That is why. Right. So one more test I'll add over here. Let me share you what I'm doing here so that you don't get bored. Are you looking at the Microsoft Word document? Can you see?
No, I can only see Microsoft Edge.
Oh,
Now I can see it.
sorry. No, okay. See, uh, I have made this for the ease of the students. The test that I need to add over here is the vitamin C test. To test the amount of vitamin C, reagent is going to be DCPIP. Original color is dark blue or slash blue. You don't need to boil it. Don't need to boil it. And then color for negative result is blue, which is like original color, dark blue or blue. And positive is transparent. Now, let me save it first, and then I'll be telling you what is all this. Basically, this is the document I made so that the students don't feel any more difficulty in memorizing all these tests. Like, you can simply see here. Okay, I'm taking you back to the Microsoft Edge. Okay, now look at here. We have a document here that would tell you about all the tests that we perform for different kinds of molecules. Like you can see, the first test that we perform here is the iodine test. And simply we perform, you read for yourself, what are we doing in this test? What are we doing in this test?
Finding the presence of starch. And so when reagent is iodine and potassium iodide, and the original color is brown yellow. And no boiling, color for negative result orange yellow, and color for positive result blue black.
Basically, what's happening? This whole thing is describing almost everything what you need to do in testing the iodine, in testing the starch. Just what it is not telling. It is not telling you about how much volumes you need to add. Like if you have a starch solution, you would add 5 ml of starch solution and 3 to 4 drops of iodine. It's just not telling you about the amount of volumes that are required to be added, but overall, this particular test is basically telling you what you are supposed to be doing in the whole experiment. So that is what this means. Then you have another test, and that is the Benedict test. The Benedict test is the only test here in which you need to uh, boil. If you see the columns properly, you'll see that you need to boil. The only test in which you need to boil is the Benedict test.
Okay.
Right?
Yeah.
Then comes the Buret test. Now remember that although these tests are going to be used in the P6, these tests are going to be used in P6, yet you need to understand them. Sometime the teachers give the P6 questions in the P2. I'm sorry, P4 or P2. They might ask you that what would be the result of this experiment. They might ask you. So in that case, you need to tell that what would be the result. You getting it?
Yeah.
So P1 or maybe the P2 or maybe P4, they might ask you that what would be the result of this particular test and how you need to perform it. So read the tests for yourself first, and then we'll be discussing them further.
Okay.
Not the table that is below.
Okay, yeah, I finished reading it then.
So always you try to remember the color differences. When you remember the color differences, and when you remember the color, the result, the change in color, when you remember the change in color, you will always be able to answer the questions very easily. Got it?
Yeah.
Okay. All right. So I'm just trying to arrange this document because it okay, I'll send you this after the class. All right. So this would help you a lot in remembering different kinds of color changes, and ultimately giving you a very good idea how to answer the question, especially if it comes in P2. Okay, let's try. Maybe there is some kind of question in P2. Oops, the past papers. Topicals. You were purchasing the account. What happened? Have you done it?
Yeah, I've done it.
Which one do you think is more beneficial? Like the Save My Exams or this? Save My Exam also has subscription. I think you might know.
Yeah, I have saved my exam subscription. But the
Which
thing
one is
is,
more useful?
save my exams problem is like very limited amount of questions.
Mm-hmm.
There's only like 5 or 6. They don't put a lot of them. This ExamMate has all of them.
Like it seriously has like limited questions even when you subscribe it?
Yeah, but like they only select like 5 questions per difficulty level. Especially when I'm doing physics. That's why I I use
Yes, yes,
examination.
yes, yes, yes. For each difficulty level, there are 5 different questions.
Yeah, which is
I
very
now
limited.
remember what you were saying. Well, that's not good. Like if we have, like obviously I have the subscription for this website. I was thinking because this website has one limitation. I also teach Edexcel, so it does not have Edexcel topicals. It does not have
Uh,
Edexcel
I
topic.
think you could try. There's another website.
Hmm.
It's called Mark Hint.
What's the name?
Mark Hint.
Tell me spellings. M.
MARK
Hmm.
Hint dot IN.
Dot in. Markhint dot in. Okay.
Just check if they have what edX, because
Past
if you go
papers,
to
topic.
topical paper
Hmm.
and if you're bored,
Edexcel
or
video.
only A levels.
Okay. IGCSE, they are not okay. Let's see. WBI 11 or 01. Okay, let's see. 11. Unit 4. Variant, any variant. Topics. Okay. Let's see
For
this one.
variant, you have to select all of them if you want any of them.
Okay, so we can, alright. Maybe May, June, October, November, any. Difficulty level, medium.
And you have to
Let's,
select the years.
hmm. Okay, we still have to select more.
You have to just select all
Okay,
the years you want. Yeah.
unit 4, sorry, unit 4. Let's say unit 4. I have done all of them.
Yeah.
Get questions.
Oh,
It does
yes,
not have.
some of them don't work. Yeah, never mind. For edXL is quite limited, I think.
It actually is limited, but the thing is that it's quite you can say
Different
the
than.
students from the no the students from the you can say Middle East countries, they selected a lot. So that is the issue. Sometime we need the past papers, and anyways, let's see what I can do for that. Save my exams might have the topical past papers for the Excel. That is why I was trying. Anyways, so let's do this question. Let's see what you can do here.
Okay, so this is C.
This is C. Exactly. This is C. Okay, let's see.
Okay, it's A because it doesn't have nitrogen.
Well done. Which was correctly pair of molecules with smaller molecules used to make them. Okay, let's see.
I don't know what this one is.
Okay, large biological molecule. Which row correctly pair large molecule with smaller molecule used to make them? Like you have to correctly match them with their subunits. So cellulose is made of glucose, yes. Oils are not made of amino acids. Oils are made of
Oh, okay, yeah.
fatty
Oh, I
acids.
understand. I thought it was just like the combination. Yeah, so yeah. Okay, so three,
Hmm.
three is correct. Um, Cellulose, I think.
Yeah, cellulose,
Yeah.
starch, and glycogen all are made of glucose.
So one and three. So B.
Yes, cellulose is a polysaccharide. We basically have read three polysaccharides. One is cellulose, starch, glycogen. Cellulose, starch, and glycogen are three polysaccharides, but their subunit is glucose only. Like subunit is same. Okay. Next, all right. Okay, what should be present against the protein?
Amino acids.
Exactly. And what should be present against the oil?
Um, Oil was, um, wasn't it if you hydrogenate? No.
No, 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 no. Oil and fats. What did I tell you? The lipids are made of fatty acids and glycerol.
So what would be for
Remember?
oil? Mm,
Fatty acid glycerol. Lipid, if a lipid is liquid, it is oil.
yeah,
If a liquid is
solid
solid, it's a fat.
fat.
So it means every lipid is made of fatty acid and glycerol. It depends on its situation. It depends on its position of matter. If it is liquid, it is oil. If it is solid, it is fat. But against the oil, there should be fatty acid and glycerol. Okay, let's see this one.
Chemical elements in protein. Um, Carbon, hydrogen, oxygen, nitrogen. A. All of them.
That's very good. Okay, now let's do this one.
When heated with a,
When heated with reducing sugar.
so that means a, what do you call it? A negative result. So
Why?
blue to on reducing.
This is a procedure. This is a procedure that you have to boil it with the. Okay, one thing
Oh
I
yeah,
told
yeah, yeah,
you.
yeah, 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 yeah. Okay, I remember. But you have to.
I said in the Benedict we have to boil. Benedict actually is for reducing sugars, but at this level we might not need to be aware that what are reducing sugars and what are not. But simply putting it, if you say that Benedict is a test for reducing sugars, uh, that still the procedure would remain same. You would have to boil it. So now, what would you say? What would be the color?
So the original color is blue,
Hmm.
and what I'm confused is with from the question, how am I supposed to tell if there's a higher presence of glucose or there's no? Oh, it's just
Okay.
presence of glucose, right? So it has to be blue
Hmm.
to red.
Blue to red is the presence of reducing sugar or the glucose. Yes, exactly right. Red to yellow is wrong because the original color is
Yeah.
not red. Similarly, brown to blue black, obviously, this may be true for iodine, for starch, and iodine, but it is not true for reducing sugar and Benedict. Blue to purple, this result is for proteins. Okay, let's do the next one. See, I told you P2 does appear. In P2, they often ask about the food tests. That is why we need to properly strengthen it. Okay, let's see how you can answer this one.
What describes the structure of DNA? DNA structure. D, right? Double helix.
Each strand contains mRNA, totally wrong. Each strand contains a sequence of amino acids, no. Fatty acids, no. So D is right. Good. All right. Now this is a real test for just today's lesson.
Okay. Below Benedict solution and positive, like the column, can you write glucose?
Website won't let me to write.
Okay. Glucose. I'll just write glucose.
Just give them number 1, number 2, number 3 on your own notes.
Okay. Glucose 2, Biore positive means protein. 3, ethanol emulsion negative means no fats. And 4 is iodine solution, which is negative, so no starch.
No starch, yes.
Okay, what did the fruit contain? So glucose and protein. So protein and reducing sugar, so C.
Protein, sorry? Yes, protein and reducing sugars, exactly. Exactly. Okay,
What?
which pair shows the correct combination of bases? Okay. Did I tell you that?
No, I don't think so. You only told about the double helix.
I told you only about the double helix, and I told you about polynucleotides, and I told you that the polynucleotides have to twist around each other, right?
Yeah.
Okay, this might be asked according to the another chapter. Okay. All right. So yeah, that's a very good question.
So amino acid.
Yeah, amino acid is the right answer. Okay, so let's move ahead. Hold on. Yeah. Okay. Yeah, that's a very interesting question. Let's see.
Doctor tested the urine of 4 patients for both glucose and protein. The colors of the urine samples after testing are shown in the table. Which patient's urine contained both protein and glucose? Okay, so Biore solution, if it needs to be purple to be positive, so presence of protein in C and D. So A, B, C, D. So C and D has protein. B, B. B. Color after testing with Benedict solution. So brick red means that there is glucose. So glucose, glucose, no glucose, no glucose. Okay. Which patient's urine contain both protein and glucose? So D. Wait.
Color after
Sorry.
both protein and
No.
glucose.
Yeah, no. Yeah, C. Hello.
Yes, yes, yes.
So
Did you
C.
tell me the answer?
Yeah.
Okay, sorry. Might be a signal glitch. Okay, let's see.
Which statement about the structure and DNA is correct? Uh, is it D? Because I don't know about the bases.
Okay, six type of bases, no, wrong. It only has four types. Three strands, wrong. Made of chain of amine, wrong. So it's D. Only four types of bases are there in the DNA. Those are basically adenine, guanine, cytosine, thymine. We might not have discussed that. Mm, okay, let's see. I guess that's not a question to ask basically. Basic units of starch and glycogen molecule. That's very clear. Let's find any other question. Again, the constant question constantly repeating. What is happening? Okay, which element is not found in proteins but not in carbohydrates? A very straightforward one.
Which element is found in protein but not carbohydrates? Carbs, elements. Carbs elements is CHO, right? Carbon, hydrogen, oxygen. So nitrogen,
Yes,
C.
exactly. Let's try any question from paper 4. Let's see if we can get any question. This is obviously the same one. Okay, let's see. Yeah, that's an interesting question I see as B part. Other than, I guess, no, you can only fill first two columns. So just try filling first two columns.
Okay, so large biological molecule, products of the breakdown of the large molecule. Um, oil, oil. I'm still confused. Oil is made from lipids, right? Lipids is
Oil is
the
made from fatty acid and glycerol.
okay. So fatty acid and glycerol.
Mhm.
An enzyme that catalyzes the breakdown of the large biological lipase,
No, just do the first. Yeah, that is lipase. Yes, you may do only
glycogen.
first two columns.
Glycogen is it glycogen? Is glucose? Is it glucose?
Yes. Hmm.
And protein, amino acids, and yeah, I don't know the enzyme.
That is basically protease. Okay. What about uh the third row?
Protein, amino acids, and then enzyme. I don't know. Or are you talking about the
No, no.
third row? Third row.
I'm talking about third row.
Maltose. It's not in the notes anywhere.